Welcome, everyone, to our next edition of Research Aromatica. My name is Aaron Sorensen. I'm excited to introduce our speaker today and the topic. A good friend of mine, I'm excited to hear what she's got to say today, Dr. Anjanette DeCarlo. And Dr. DeCarlo is distinguished as the only female scientist who has worked exclusively on the ground on locating where trees grow in Burkina Faso, Ethiopia, Oman, Somaliland, and Somalia. She uses a dual approach of interviewing communities as well as taking samples and running forest health analysis. She founded the Save Frankincense initiative and is currently on the advisory board of the Global Frankincense Alliance. She is currently the senior sustainability scientist at the Aromatic Plant Research Center, focused on sustainability of aromatic and medicinal plants. Dr. DeCarlo received her doctorate in natural resources and environment from the University of Vermont and has worked on post-confident evaluations of natural assets and environmental projects from all over the world. Dr. DeCarlo and I have had the chance to travel together to many different locations throughout the world, doing a variety of research. She's a very good friend of mine and very knowledgeable in each field that she has studied. And I'm excited to introduce her today and turn the time over to her for her presentation.
Great, thanks, Aaron. Hi, everyone. It's amazing to see you, Franz and Shelley and Linda and Jillian and Stephen Johnson, Ahmed Didi. All of you have been such an amazing support to move this research forward on frankincense, and it's just amazing to see you all here today. We've got a really international group, and so I'm pretty excited about that. I'm going to go right ahead and jump in and get the slides up. We are completely live today, so that's pretty exciting to be doing a live webinar. My first time doing a completely live webinar, no pre-recorded sections or anything. So here we go. So here today we're going to talk about the future of frankincense, and we're going to focus on in situ propagation. And what that means is propagation at the source or on location where the species naturally grows. The future of frankincense is cooperation. The only way that we as people will come together and save frankincense is if we cooperate across divisions, across boundaries, maybe even across competing entities. But really the way for us to come together scientifically as well is to recognize the incredible value of local knowledge and local stakeholders, the people who actually live with these trees, and to combine that with Westerners and outsiders and people from China all over the world, and science, to where the breakthroughs and the discoveries really happen. That's the sweet spot in the middle there, where the breakthroughs and discoveries are happening. So I wanted to quickly show you, and just before we get into the propagation techniques, some of the overview, I want to show you some to get everybody on the same page, because we have some people who are incredibly knowledgeable about frankincense and some people who are coming to it new. This is a map of the Boswellia range where many different species of frankincense grows. And today I'll be showing you pictures and focusing on the areas where those two red circles are drawn. And that's Oman, Ethiopia, Somaliland and Somalia, and Burkina Faso. Frankincense trees have been tapped for over 6,000 years for the precious resins that they produce. Humanity has had a love affair with frankincense for a very long time. In fact, it is considered to be, by some academics, to be one of the longest continuously traded commodities of humanity. And so we really have had a long-term relationship with frankincense. And so everyone knows the resin is part of the tree's immune defense system. So when the tree naturally gets a cut or a branch falls off, the resin is excreted to cover that wound. Or if a long boring beetle gets into the tree, it gets stuck in that resin and it can't penetrate and kill the tree. So the resin comes out, and if we're collecting it for humans to use, it's scraped off of these cuts. And on the left there, you see a sakura tree being tapped in Oman, and on the right is a papyrifera tree being tapped in Ethiopia. I think this is such a beautiful picture. This is a Feriana tree. They're very distinct in the way that the resin seeps from the tree. It almost looks like long flowing hair coming out. When the resins come fully out and when it's time, the harvesters will scrape the resins off the taps in cycles. And if it's being done sustainably, on the last round, they will leave the resin in place so that it can form that protective cover over the wound and protect itself. This is probably one of my favorite resin shots. It was the biggest piece of Carteri that myself or Stephen had ever seen, that I'm holding there on the right in my hand. That's from a newly tapped tree, a tree that had never been tapped before. Resins in general are sold for incense, oils, and extracts. Current global demand is very high for certain species. Frankincense can be found burning as incense in places of worship. Many people have memories or still go to places of worship where it's burned as an incense. It's used in perfumes, in anti-aging products, essential oils, pharmaceuticals, herbal and aromatic products. The resins are often distilled into oils or extracted for the heavier compounds, such as boswellic acid, which is quite a well-known anti-inflammatory. So thanks, Julie, for asking about the biodiversity and some of the other species that grow around frankincense. You should be getting a handout that has at the bottom a list, a very short list, but of some of the species that do grow around different types of frankincense. I'm going to do this very generally and just show you some of the stars of biodiversity. But what you're looking at right here is a drone shot from transect lines that we use drones to fly. And you're right over the Almado, which is in Somaliland, Somalia. It's a beautiful shot. We love it. We wanted to show you drone footage, but we're not able to do that today. But this is such an amazing shot, and you're seeing some of the different species that grow. And let's take a look at some of those a little more closely. So, of course, one of the big stars is Chamaephora myrrha. And the resins from myrrh, this is a young myrrh tree. They're as well known in many circles as frankincense. Right? Frankincense and myrrh were both brought to baby Jesus together. And so Chamaephora myrrha and frankincense are often used together in traditional medicine for the healing effect. And some people even speak about them as being the yin-yang of each other medicinally. So the Chamaephora myrrha can sometimes grow very close to frankincense trees in the drier regions or up on the dry areas above where the frankincense trees grow. There's also a lot of wildlife. There's more wildlife than I have time to put in this presentation. These are wolf tracks at the base of a sakura tree in Oman. I was pretty excited about that. And on the other side is actually something that's heartbreaking, which is the trafficking of cheetahs that happens a lot through the growing region in Somaliland and Somalia. And this amazing woman cares for them as part of a GTZ funded project, which is now expanded in Somaliland. And they do have rehabilitation centers for the cheetahs to be able to care for them when they are confiscated. And this woman she gives her whole life to this. This is pretty much what she does. These are her babies. And we were pretty lucky to spend a few days with her. The other star is Dracaena, which is in some places quite a rare species, growing here in the foreground. I'm going to turn on this whiteboard for a moment here and show you. Here it is, right here. This is the Dracaena umbut right here. There's quite a lot of these in the Almado in Somaliland. On the other side, on the right-hand side there, you're looking at in the front, that's a frankincense tree. And in the back, you have a Vitellia, formerly known as acacia. And way in the back there, that's another Dracaena. Okay. Growing above the frankincense trees in Somaliland is a remnant juniper forest. And the reason why I say remnant is because a lot of those trees have been deforested over many, many decades for the timber. And these majestic trees are also being cultivated in a nursery called the Dalo Juniper Nursery. It's just above the frankincense trees. And if there's a juniper nursery, we certainly can have a frankincense nursery, which we're going to talk more about. And in fact, there are frankincense nurseries popping up. But it's very exciting to see a juniper nursery to do reforestation of the juniper trees in this part of Somaliland. And that's run by an NGO called Candlelight. Two other trees that are just striking. One is Aloe eminens, which is it's an endemic species to the Almado. It only grows in that area of the world. And on the other side, the Conocarpus, which, I mean, we were just amazed. And just a few hundred meters up behind Fariana trees were growing. So they grow in close proximity. I wouldn't say they're in a symbiotic relationship, but they're quite close. Here's just to give you an image of a papyrifera forest in Ethiopia and a wadi in Oman. These are sakura trees growing in a wadi in Oman. So let's get to some of the challenges as to why we consider propagation as part of the future of frankincense and why that's so important. Well, there's a lot of challenges facing forests and trees globally. We all know that. In the case of frankincense, almost everywhere the trees grow, there's food and water instability. People are living either with food instability or water or both. There's poverty, structural poverty, which, as most of you know, is a form of violence and can actually exacerbate problems for people and their natural resources. But there's also conflict as well. So the border of Ethiopia and Eritrea, which is now definitely a lot better than it was in the past, but there's a lot of conflict, and that's where this picture is taken, where the bullet cartridge there you see inserted into a papyrifera tree. Some of the other challenges are overgrazing. This is in Oman. This is a tree completely overgrazed on the left. All the buds were eaten off by camels. And on the right-hand side, you see a sakura tree that's just outside of an enclosure, protecting the sakura trees within the enclosure. This one's just outside of it, and you can see how high up the grazing's happening, and that's a real indication that that's camel grazing. Land conversion, fire and burning for population growth. People need these lands to grow crops. This is Ethiopia, where this picture was taken outside the Asosa region. And of course, overtapping, overuse of the resource by either putting too many wounds, doing it at the wrong time of year, doing it too often, not allowing the trees to rest. And so that's just very brief of some of the challenges so that we can now really get into the heart of today's talk, is to focus on the solutions. We've made a lot of observations from nature that inform propagation, things that have really helped us to say, oh, about the trees. And not just myself, but people like Salah Ajib, who's been propagating frankincense for over 25 years in Oman doing this, have been watching and looking at how the trees grow in nature to understand how human propagation can move that along, or how we can do it faster, how we can bring the trees along faster. Well, one thing is that the trees do grow on a really interesting geology, which is these lava flows that come over the limestone escarpment. And so you'll often see the trees growing out of volcanic rock, and sometimes you'll see them growing on the limestone, and sometimes you'll see them growing on a mix. So here's what I mean. Here's volcanic rock. And you see there a little natural hole in the rock has collected some water and some organic matter. And then amazingly, Boswellia trees have the ability to literally grow out of these holes in rock. It's amazing to me that this tree has the ability to do this and also behave more like a regular tree with real roots. And I'm going to show you what I mean by that. Fog and rainy seasons, distinct rainy seasons, are also something naturally observed. Frankincense trees are often fed by fogs and condensation that we've seen in Ethiopia and parts that we've been. We see a lot of condensation, like that early morning dew. But in the Almado facing the Red Sea, that you get these fogs that are really, really characteristic of this area. And those fogs feed water to the frankincense trees, as do the discrete rainy seasons that happen twice a year, but absolutely do not deliver large amounts of rain in this part of the world. So back to these trees that grow just on the rock. What we have observed is that the trees that grow on rocks are usually smaller, and they're much more susceptible to being blown down in storms or to being overtapped, because they're really literally growing not with roots that move out, but a root ball. They kind of almost sucker themselves to the rock. It's an amazing ability that frankincense trees have, but they are more susceptible when they grow like this, which many of them do grow like this. Now, what we've seen too is that for a tree to get big, or for it to become a mother tree, so to speak, that it needs to be able to get its roots off of the rock and into soil. And so that's what you see in the smaller circle there. Those are the roots of this mother tree getting off the rock and going down into the soil. And this is the biggest carteri tree that I've ever seen. And the distinguishing characteristic here was that it was able to get its roots off of the rocks and into soil. So all of those things inform scientists as to how propagation can be sped up. Okay, so let's get into it now. Propagating Boswellia, we're going to talk about two types. Propagating Boswellia from seeds or from clippings. Now, there are other ways to do it. There's tissue culture, there's some other ways, but really these are the two central ways to propagate Boswellia. I'm not ruling out tissue culture. I'm not saying tissue culture is a bad idea. It's just, this is what we have to work with right now that people are doing right now. And so you can see this is a real picture of a seedling propagation center. This is a picture taken from Oman. But let's talk a little bit for a second about the myth. So the myth is that the trees can't be propagated by humans. And I've heard that from many people in the communities that they don't really feel like this is possible. And we'll get to the second myth that accompanies that. But that's false. That is false. These trees can be cultivated by humans. And in fact, I'm going to show you lots of different places where it's been successful. So seeds have an advantage because plants grown from seeds are great for reforestation. They develop their roots as they're developing their stem and their leaves. And so that makes them have hardier roots that can be used to be planted back out into the wild where they will not be cared for. However, some studies have also shown that viability can be low with seeds, and that may be linked to decline in tree health. So to do that, to collect seeds, you want to take the fruiting bodies, which you can see here in this picture. This is a papyrifera tree. It has beautiful pink flowers that after the flowers come and go, then the seed bodies, the fruiting bodies emerge. And inside those pods, I'll show you some pictures here, are the seeds. You want to take these fruiting bodies from the tree and not from the ground. That's a really distinguishing factor. The pods should be reddish brown when they're taken, and then they're crushed and you remove the seeds. And you can see me pointing at a tiny little seed there, and there's usually about 4 of those in each pod. Here's some Dalziellii seeds in Burkina Faso, and you can see these are too green. So this is an example of seeds that were not mature enough and were not ready to be taken off the tree. we experimented with these. They weren't viable yet. They weren't ready to go. And so if you're a landowner, because I know we have some people on today who own trees, who want to propagate more trees, who are really interested in this for on-the-ground efforts, as well as NGOs, academics, and companies who are all looking to do more propagation of the trees, you definitely don't want to take them when they're green like this, because you won't have viable seeds. They're not mature enough yet. So May and June are the best months for sprouting seeds, specifically in Oman and Somaliland. Somalia, I would think it's about the same across West Africa, and probably for Ethiopia. I mean, Franz might be able to say yes or no to that. Because I'm giving the slideshow, I can't follow the chat till the end. So pardon me if you're asking something or saying something and I'm not acknowledging it, but that's why. So, but please talk to each other in the chat. So to sprout seeds, you only need about 5 to 10 centimeters of nice wet soil. And then you sprinkle the seeds in that and cover it with plastic. And the reason why you're covering it with plastic is to increase the humidity. Remember the fog picture that I just showed you a couple minutes ago? These seedlings like to, sprout in high humidity. They need the high humidity to sprout, and that's part of their natural environmental ecosystem, is to have that fog to sprout. And so you cover that with plastic without the plastic touching the soil to increase the humidity. If you have high humidity, then you don't need it. But most of the places where frankincense grows are quite dry. Maybe in Ethiopia, you might not need it. As soon as the first sprouts start to happen, you can remove that plastic. And so you see a little baby sprout. Look at that. That's just amazing. In the lower circle there, that's a sprout that's reached its second sprouting stage. And they're just so cute, those little sprouts. The ones in the larger circle are certainly quite a bit older. At about 10 to 14 days is when the second sprout occurs, and those babies can then be moved to pots or bags. Now, because again, we're talking about propagation in the places that the trees grow, you have access to the native soils. You have access to the soils that the trees grow in naturally. What we also know is that if we mix that native soil with compost, that will help the baby tree grow faster and stronger and develop its roots more effectively. So we're trying to move the process along. Remember, by intervening and doing human propagation, we are looking for ways to naturally and sustainably increase the regeneration rates by quickening the propagation. So compost and how to compost in these areas is a whole other webinar class on its own, but there are ways to produce compost from dried animal manure, from dried grasses and clippings that can be done with very little inputs and are appropriate for communities in developing countries. So you mix the 40% native soil and the 60% compost and leave them in the bags for 6 months to 2 years. Now, that might seem like a long time, but actually it's really not. To get this really good root system going, you want to let them stay in the bags for as long as possible. Here are a few sakura trees planted in a farm in Oman. They were grown from seed and they're now a few years old. You can really tell that these are grown from seed because they're so bushed out and they have many different trunks. This is the UNESCO World Heritage Site in Oman, and everything you see there are sakura trees that were planted in an enclosure, and they were mostly from seed. I think, in fact, if not all from seed. Now, some people are interested in today's topic because they want to plant frankincense in cultivation where they will be caring for the trees. Some people are interested in reforestation and how the trees can be grown in nurseries and then replanted into the wild. they're two different things, but some people want to do both, right? For reforestation, seedlings, again, are preferable because they're developing strong root systems as they're growing their trunk and their leaves. Here's an example of a seedling that's been grown in a bag for a year, and it's going out. This was actually pictures that were just recently taken. It's going out for reforestation in Oman. To do that, the day you're going to do that, you water the tree really well. And at a cool time of day, because you don't want to shock the tree, you dig a big hole and you put compost at the bottom. Now, why are you putting the compost at the bottom? Well, you're encouraging the roots to grow down. So you want to put the compost, and in fact, with most plants in your garden or in your farm, you do the same thing when you're first planting them, especially for trees. Put the compost at the bottom, and that encourages the roots to go down. After you fill the hole with native soil, water that tree very well and leave a small depression so that the tree can collect rainwater and runoff more effectively. Because after that, these trees will get no more human care except for maybe to monitor their success. Oman really is an example for the rest of the world in terms of propagation of frankincense. A lot of what I've learned is from people in Oman, and Salah Ajib has taught me so much. I'm so grateful to him. He was the project planner and the teacher of propagation for the Ashore enclosure. And you could see that the government of Oman, the Sultanate of Oman, and their ministries in 1995, after experiencing some decline in their sakura trees, which today we're not getting into the reasons why, but they had some decline in their sakura tree populations, and they started to take action in the 90s to assess the situation with the trees and really went forward with propagation and putting seedlings into enclosures and growing them for many reasons. One, to be a seed bank. So you have incredibly good specimen trees growing in an enclosure, 6,000 of them now as of 2014. And to also take genetic material from mother trees, trees that are very strong, that are coming from deteriorated areas where the trees might have been in decline, and move them here for a genetic seed bank. I've also seen sakura trees cultivated right in the United States of America. These were the former mini tree garden, which has now moved its operations because if you get frost at all, you can't grow Boswellia trees. And this is a sakura and a Carteri synonym sakura growing in Arizona. This is about 4 years ago. And these trees actually were moved to a warmer climate. Here's also just a few more examples of seedlings that have been dispersed around the world. When I first started working on frankincense over 10 years ago, my colleague Marta Cironi got a hold of some seeds and planted them in the greenhouse at the University of Vermont. And we went back about 6 months ago, and we're really pleased to see them growing. They were struggling a little bit. The greenhouse manager said they really needed to make some changes to be more conducive to the Dalziellii, but it was growing there, and it was in its dormancy phase. The other one is seedlings grown on Professor Amade's desk in his lab in Burkina Faso. And of course, for many of you in the UK, if you've ever been to Kew Gardens, there's a couple sakura trees growing there from seed, and they're surrounded by aloes, which is another species of plants that grows near frankincense. So the other way to propagate frankincense is from branch cuttings. Now, branch cuttings are technically clones of the tree that you take them from, right? They're genetic clones from that tree. And why branch cuttings work for frankincense and other tree species, there's other plants that you can do this with for sure, is that plant cells, unlike animal cells, are totipotent. They're totally potent, which means any one cell can grow into the entire plant. It's simply amazing strategy that plants have to keep themselves alive. And in fact, they predate us on the planet. And so this is why clippings can work. Clippings can be fast growing. They take longer for root development, but they do extremely well in cultivation where they'll be cared for several years. Taking branch cuttings is sensitive. You have to do it at the right time of year, which is at the end of the dry season, just about when they're about to resprout leaves, because that's when the tree's chemical signals are telling it to resprout and regrow. How you take the cutting is what can be very sensitive because of the fact that you don't want to take multiple cuttings from the same tree. Now, why? Well, in a lot of areas these trees grow, this is an occulta tree growing in Somaliland, there's a pest called hare, and that's the Somali term for it. It's a long boring beetle. And when you cut branches, you essentially leave a wound that is potentially dangerous for pests to get into the tree. And so in some locations, people are not comfortable to take too many branch cuttings. They do it very, very carefully. And they then take branch cuttings from the branch cuttings, which I'm going to show you. So that is a drawback of branch cuttings, is that you are wounding the tree. You want to take a branch that's young, maybe 1 to 2 years old branch, and you want to cut at the node, which is where we've circled there, which is to cut right at that node. You don't want to just cut the tree in the middle of a branch, and you definitely don't want to cut the main stem. After you've taken that 1/2 meter piece from the tree, you can then cut that into 25 centimeter pieces. As long as they're about 1 to 2 diameters minimum, then you're good. You want to keep them in the direction of the growth of how the branch was coming out of the tree. And one of the ways that humans can speed things up a little bit is by dipping the base of the branch cutting into root hormone. And again, then placing into bags that are 40% native soils and 60% compost. Good drainage is essential. You want to make sure that the roots are never in standing water. Good drainage is essential. It's best to place the pots or the bags on gravel pads. And you can see here, these cuttings here in Oman are on a gravel pad, and below in the circle, I look very happy, but those clippings actually failed. That's in Burkina Faso. We took clippings. We just tried. We took them at the wrong time of year just to see what would happen, and they didn't grow. So we've learned as much from our failures as we've learned from our successes. And for sure, you have to take the clippings at the right time of year, or you don't really get the kind of success, at least with Dalzielii, that we would like to see. Here's a little initial growth at the base of a clipping there, and that's very exciting. When you start to see that happening, you know that your clipping is going to make it. It's going to happen. You have to keep them under shade cloth, and that green effect there is the shade cloth in pots or bags for up to 6 months to 2 years, watering every 3 to 4 days. Here's another shot. This is in Somaliland. This was at a sheikh school where they were experimenting with taking clippings and growing them in pots, and they were very successful. Sacra and Carteri, because Carteri is Sacra, seem to do incredibly well in clipping propagation. I mean, the results, even with suboptimal conditions, like maybe not having compost, we've had success with being able to get root clippings to take. People can even do the root clippings with Commiphora myrrha. So this is an example of myrrh clippings. And here's your clippings really coming to fruition. They're just leafing out. They're getting strong. When the roots are developed, the young trees then, the clippings, can be planted into the ground, mostly in cultivation, nursery, and farms. Because they are clippings, they're going to need longer-term care. Here's some clippings that made it for a year in their pots and then were planted into the ground. This is a nursery in Somaliland, and it's about 300 to 400 trees currently growing really nicely. You could see the depressions there for the water collection. And of course, they are being watered by hand here in this case. This is one of my favorites. This is, the men are standing around the base of a papyrifera tree that was cut, a very large cutting. The community took a very large branch off a very large papyrifera tree and planted it, and it took. It was amazing for us to see. I mean, you know, one of the men here is at least 6 feet tall, if not more, so you can really see how big that papyrifera tree. But because it's in the community, they water it, so it does get care. And they also planted many others that were being used as fence posts. And in this area in Ethiopia, in Asosa, the trees are able to take because they don't have the grazing, some of the grazing problems that eliminate young trees. But this clipping was too big ever to be grazed, and it just was a pretty amazing thing to see. They were not tapping it because they felt that the root development was not sufficient to do that. When you do have your clippings planted into a farm, you are going to have to water them for up to 4 years. So you could see the spacing is, the optimal spacing is 3 by 3. If they're going to live their entire life in a farm and then be tapped in the future, they also have to be watered in this pattern for the tree's health. And also, if you're growing them in a farm, you're most likely growing them to tap them. And so you have to follow this watering regime to be able at the end to get trees that you can tap for resin. Here's some sakura trees that are a few years old, have been grown in cultivation. And this was something that we just experimented with. Remember those holes I showed you at the beginning where the water had collected in the lava rock? We put a clipping in one of those. One of the teams put a clipping in one of those, and it actually took. Didn't expect that. Didn't expect that at all. And so we're starting to see how resilient this tree really is. Okay. We're almost at the end here, and this is the myth that probably some of you were thinking in the back of your mind is, well, if you can grow frankincense tree, if humans can grow them, that's all well and good, but that it won't give good quality resin. It won't give the kind of resins that we get from the wild, the complex chemistry and yields that the industry is looking for. Well, that's both true and false. This picture is a tree that's been watered and tapped its entire life. It's behind a mosque in Goodmo. And the reason why this resin is not very good quality is because the tree is regularly watered for its entire life. And why is that a factor? Well, if you take a look here, the top picture, these were taken the same day or a day apart. The top picture are trees growing in a wadi in Oman, and the bottom one is growing in a nursery and being watered. So to be able to get the complex chemistry, the trees need to have some stress. They need to be a little water stressed. And so after that 4-year period, no more compost, no more fertilizer past the first year, and following that water regimen, you can start to tap the trees. But in the first year and the second year of tapping, that resin may not be high quality, but by the 3rd year of stressing the tree, by the 3rd season of tapping, you should be good to go. I'm going to end with this picture. This is a farm in Puntland. They went through the process that I, kind of very basically went through, and you can see how well these trees are producing resin, and the resin quality is phenomenal from these trees. In fact, I've looked at the GCs of this resin, and it really doesn't, you can't tell the difference from it or a tree grown in the wild. So I just want to say thank you to everyone. This was a very brief introduction to how to cultivate Boswellia. But we want to start making this information open source for people who are landowners and have trees, and or academics, NGOs, and companies who want to propagate these trees all over the growing areas that they naturally exist. So thank you very much. I'm going to move over to the chat here so I can see. And I believe Aaron is going to come back on for Q&A.
Hey, Anjanette, great job. I learned a lot. Thank you.
That's great. So
there are several questions on there. I don't know if you get a chance to look there. And Denzel was going to.
Oh, great. Is Denzel with
do
us?
An introduction of. Yeah.
Yeah. Can we bring him up for a minute?
Yeah, he just needs to click on speak there. Let's see, I'm helping him in now. It'll take just a second to have him come in. But we'd like him to talk a little bit more about the Global Frankincense Alliance.
Yeah, that would be great. We'll bring him up. So while we're bringing him up, I can just say a little bit about the Global Frankincense Alliance. Is he coming on? It's a recently formed organization, and out of the last two frankincense conferences, and the last one being in Cyprus at the World Congress on Medicinal Plants, groups of experts, companies, practitioners, landowners, tappers came together and really supported the formation of a global frankincense alliance. So there's groups that focus on smaller areas or research in specific zones. And we really have seen that to work on protecting and saving frankincense at the broad scale, we need to work at a global scale, and we need to work across country and species to be able to do that. And so the Global Frankincense Alliance, I'm on the board of advisors with a group of amazing scientists. Dr. Bongers is with us today. He's also on that board. But it's answering the call for bringing together the research from many, many different countries and many different people, and supporting those efforts to conserve frankincense. And so I know Denzel is really a supporter of propagation of frankincense. He's worked for many years in Oman as well, and he's friends with Salah. So propagation is near and dear to his heart. And so I don't think we're bringing Denzel up, but I think I covered a lot about it. And you can certainly visit the Global Frankincense Alliance webpage to learn more about it.
Denzel, are you in? All right. Well, we'll keep trying there.
Okay.
But maybe if we look through some of the questions here, there's some great questions asked. One from Jay Fry: once the resin harvested, how long does it remain edible or usable?
Oh, that's a great question. So I have seen frankincense in museums on display. Frankincense doesn't, quote unquote, go bad. Aaron, I think we tried in the lab to grow some bacterias on frankincense, right? And we couldn't get it to grow anything. Am I remembering that correctly?
Yes, and we've got some that aren't, we don't have any from tombs of ancient Egypt or things like that, but that are at least a decade old. And still, as you break them open, they have the characteristic aroma of frankincense there. And the yields do definitely go down as it continues to evaporate over time, but it is still there. The chemistry seems to remain fairly consistent. It's hard to say when it expires, if you will.
Yeah, we had a stump, a Dalzielii stump, that had been cut down about 12 years earlier, and the stump was still exuding resin. And the owner of that land was so intrigued by that. And when he sent us some of the resin, we could break it open and still smell the characteristic smell of Dalzielii. So it doesn't really go bad. And that's partly why it was used to embalm mummies, with myrrh, along with myrrh for the ancient Egyptians, because it's such a great preservative. Yeah.
Another question I see on here: does the age of the resin from original harvest to the day it is used to isolate oil, is there any changes in the oil composition? And I'll, if you want to comment on that,
No, go
Yeah.
ahead, Aaron. That's a good one for you, actually.
But I have seen that we've with the different species of resin, you can when it's very gooey, still wet, and when it's fresh, and the major impact is on the yield, where it's still got the maximum, if you will, amount of oil in that resin there, and a lot of the lighter components like the alpha pinene, and many other of the just the much lighter alpha pinenes and things like that. Beta pinenes are much more are higher concentration, alpha theugene too, and those will dissipate more easily, more readily over time. And so yes, there is a slight change in the, I guess, the ratios of the composition, but the components seem to remain the same, but major impacts on how much yield there is in the oil, depending on when it's harvest time versus how much time it's allowed to dry and sit.
Yeah. I see a question from Brian Lawrence about environments where Boswellia will not grow well. And I just mentioned very briefly that anywhere that experiences even a light frost, it's one thing that they just cannot tolerate at all. And so Miniature Garden used to be in Arizona, for example, and because they had one frost, everything had to be moved. It was an emergency situation to save the trees. And so they certainly can't grow in Vermont or anywhere that it gets cold, as well as, not too wet because they don't like standing water. So clay soils that would hold a lot of moisture close to the roots would not work well for them. They need drainage. And so, but the main thing, Brian, is the cold is the biggest factor. Yeah, I see Ahmed Aden is here. He's a farm owner of frankincense, and that's so exciting. We're really, we really want to work with people who own the trees to be able to understand propagation and to be able to do it. I mean, many of the elders know a lot of this already. I mean, there's elders that really do understand how to cultivate from cuttings. It's an ancient way of transferring the trees. So that's not a modern scientific discovery. It's just that we can add things to that process and speed it up and speed along the process. Let's see, were there any others here?
I did. So it was actually answered very well by Stephen Johnson in the chat, but there was a question asked about male or female trees, and you're wearing a shirt on that, Anjanette.
Yes, so I am. So the trees are monoecious, meaning that the male parts and the female parts to self-pollinate exist on the same tree. So sometimes resins are referred to culturally. We've heard this before, the male resins or female resins, but the trees themselves are monoecious. Thanks for that, Stephen.
Another question here, what is the life expectancy of a tree?
Oh,
Probably
that's
a lot of factors in that, but yeah.
There's a lot of factors in that, but I've seen some claims of these hundreds and hundreds of year-old trees in Oman. But I have no way to substantiate that or no. I mean, someone like Salah would be able to say whether or not that's a myth or a fact. But the reality is, in Somaliland at least, we never wanted to. I mean, we wanted to as scientists, but it was inappropriate. It's not appropriate to core bore the trees and figure out their exact age. It just leaves them open to hot air, and culturally that doesn't feel good to the people there. They don't really like that idea and totally understand that. It's not my favorite thing to do to a tree either. But on average, we see the trees being between about 75 and maximum about 100, 125 years old. They start producing in the wild between 25 and 40 years old is the good range for starting to tap the trees. And so, as I said, too, when they're growing on rock, they could be older than they look because they can't get too big growing on the rock like that when they're just on the lava boulders versus the trees that can get their roots down and grow into soil and grow much bigger. So the size is not always the best indicator of age. but this harsh climate and blowdowns, the trees get blown down very easily. And so, like I said, that mother tree we showed you, the community felt like that tree was about 100 years old.
Interesting.
Yeah. Yeah. Any other ones there? Sacra and Carteri are two different species, according to the to the GC profiles. So this is another one that maybe Stephen Johnson answered in the chat, but Carteri is a synonym for Sacra. I mean, Matt Thulin has, kind of waved his finger at Stephen and I and said, be careful about this, of making sure that you're being clear that Carteri and Sacra are the same species. Carteri is a synonym for it. It's the meaning that it's growing in Somalia or Somaliland. And there are chemistry differences between them, but that doesn't necessarily mean they're different species. Carteri, and Aaron, you could comment on this, it has a lot of diversity in chemistry, right? We've seen two major chemotypes with subchemotypes. We were able to separate out occulta as a new species with Dr. Thulin as well, that it wasn't a hybrid or something, just a chemical variant, but its own species. But Carteri is one of the ones that really does have a lot of chemistry diversity.
Yeah, definitely. And we've done dendrograms with Dr. Setzer, looking at hundreds of different samples of whether it's resins that we took and distilled ourselves or samples from various sources. And you're exactly right. There's quite a bit of variance or distinguish chemistry in those. And whether that's, of course, environmental factors come into play there, age of the trees, so on and so forth. Being able to see new chemicals and isolate those into new species with a colta, as you said, with Dr. Thulin, confirming that. So really fascinating. We learned so much more about this species and these trees on a daily, on a year-to-year and even a week-to-week basis that it's just fascinating. There's years of research to still do.
And we don't know exactly why the trees have so much chemical variation. Right. So I was just going through this with a colleague, and he said, you can't say that it's the rock, or you can't say that it's a lot of the things that you would normally think it is, that there are scientists doing research on why there's so much chemical variation, whether it's a fungi interaction, whether it's bacteria, what is happening here is the rock, the substrates. And we just can't say right now because we don't know yet why Carteri in particular has so much chemical variation. So that's going to be one of the breakthroughs coming in science as to why.
Yeah, and continuing to gather data so that we have as many data points as possible from different regions, and looking at the chemistry of that is going to just add more light into the why as the research continues.
Mm-hmm.
Okay.
Well, great. I think Denzel is there now. Should we try bringing him up for just to have the last word?
Yeah, I'm going to try.
We're almost out of time.
It keeps crashing there. I'm going to try and have him
gone.
get in. I've clicked on it a couple times, and he'd mentioned that I'll try again here. But he does mention in a comment there that CITES is looking at frankincense species.
Yes.
Something that you can speak much more about, Anjanette.
Thank you, Denzel, for reminding me to say something about that. Yes, the Boswellia are under review by CITES. The IUCN has in the past listed Sacra as a threatened species. So now with the increased demand and trade of frankincense, its increased popularity, and also all the other pressures, the poverty, the issues we went through, the insecurity, the conflict, the challenges that exist, it's under review to see if it should have protection for trade, and that would mean that if in fact it does pass, that there is more protections for Boswellia species, it would mean more transparency and permits, etc., to be able to show that you're sustainably tapping the trees and that you're also propagating the trees to have regeneration and to be able to trade the resins internationally. And it's very controversial. Some people are very much in support of a CITES review, and some people are not. And some people point out where CITES has been successful and where it has failed or made it harder for small companies or small farmers to be able to trade their products. And so it's an unfolding process. It was delayed due to the COVID situation, the global situation, but that will be occurring over the next 1.5 to 2 years, where a final vote will occur by the CITES at a COP meeting. Yeah.
Definitely. Certainly many more conversations to be had in that realm there too.
Yeah. Well, thank you so much, everybody.
Yeah, thank you. And Denzel did put up just globalfrankincensealliance.com, all one word, is where you can get on and see more information there too.
Great.
Well, thank you, everyone. I know we didn't get to everybody's questions, but feel free to, we can follow up with other emails, and we're happy to, this educational platform is intended to be an opportunity for everyone, experts in a variety of fields in aromatic plants, to present their experience and their years of being able to study these plants. So thank you again, Anjanette, for your time, and for everyone who participated. And we'll see you again in a couple weeks.
Great.
Thank you.
Bye.